I'm Sarah Hamlin.
I'm Mandy Lace.
I'm Ashley Bell.
And I'm Tara Smoker. So the first step that we did was we took 1.5 milliliters of our overnight bacteria and placed it in an Eppendorf tube. The tube was then placed in the centrifuge for 1 minute. The supernate, the excess material on top was removed, so just the pellet remained. And then 200 microliters of the resuspension material was added. Then we took the tube and we rubbed it on top of the Eppendorf holder to mix the pellet and resuspension material.
Next was the lysis step. So we added 200 milliliters of lysis solution, and then we inverted it 6 to 8 times. And then the precipitate is what we needed. So the cell debris was the pellet after we centrifuged it. So we got rid of the pellet, and then the liquid was the DNA that we kept.
Okay, and then in the liquid with the DNA that we had remaining from that, we ran it through a column. And the way that works is the column is filled with stuff with a positive charge. And since DNA has a negative charge, when we run it through the column, the DNA will stick to the column and everything else will run through it. And so we discarded the flow through, and then we ran a wash through the column, which kind of gets like proteins and other stuff that's stuck with the DNA, and that kind of rinses that out. And then after that, we ran an elution solution through the column, and that pulled the DNA out of the column so that our flow through was the plasmid DNA. And that works because it has a positive charge more so than the positive charge in the column, so that it has more affinity for the DNA, so the DNA will go with it. And then our flow through was just the plasmid DNA.
So yeah, after the elution solution, we had collected our DNA, and so we took it to the back room to put it in the nanodrop. That way we could quantify our experiment. And we found that our DNA solution had 21.1 nanograms per microliter, which isn't super high. That's just measuring the amount of like RNA and DNA things in it. But it wasn't super high. However, we did have a good DNA to RNA ratio. The A260 to A280 ratio was 1.86, which is indicative that our DNA was more pure than other groups. So thanks.
Thank you.